स्नेल न्यूरो एनाटॉमी से क्लिनिकल नोट्स चैप्टर नंबर 2 के आज हमें इस वीडियो में डिस्कस करने हैं। और आपको पता है कि मेरी ये जो क्लिनिकल नोट्स की सीरीज है स्नेल न्यूरो एनाटॉमी को लेकर, उसका पैटर्न ये है कि मैं चैप्टर नंबर 2 विच इज न्यूरॉन्स एंड न्यूरोग्लिया, ये वाला चैप्टर हम ऑलरेडी कर चुके हैं। सो वी हैव ऑलरेडी डिस्कस्ड, इस पे मेरी वीडियो पड़ी हुई है चैनल पर। लेकिन उस वक्त जब मैंने ये चैप्टर आपको डिटेल से करवाया था न्यूरॉन्स एंड न्यूरोग्लिया का, तो इसका टेक्स्ट तो हमने कर लिया था, बट क्लिनिकल नोट्स मैंने न्यूरो एनाटॉमी के छोड़ दिए थे। सो दैट इज व्हाट आई एम नाउ कंप्लीटिंग, ताकि आपको पूरे चैप्टर का एक बेसिक सेंस हो। वैसे भी क्लिनिकल नोट्स के बगैर वो जो टेक्स्ट है वो कुछ ज्यादा उसका मतलब रह नहीं जाता। ओके, सो लेट्स स्टार्ट टॉकिंग अबाउट द क्लिनिकल नोट्स फॉर चैप्टर नंबर 2 फ्रॉम स्नेल न्यूरो एनाटॉमी। सो न्यूरॉन इज द बेसिक फंक्शनल यूनिट ऑफ द होल नर्वस सिस्टम, और ये बात अब आपको मालूम है कि पूरे का पूरा नर्वस सिस्टम जो है उसका जो बेसिक यूनिट है, जो बेसिक सेल है, वो कहलाता है न्यूरॉन। इन द मैच्योर ह्यूमन, इफ इट इज डिस्ट्रॉयड बाय ट्रॉमा और डिजीज, इट इज नॉट रीप्लेस्ड, बिकॉज जो न्यूरॉन्स हैं दे आर यूजुअली प्रेजेंट इन G0 phase of the cell cycle, so they do not replicate. Neurons are incapable of undergoing cell division because they are arrested at G0 phase, so they do not complete the cell cycle. The neuron consists of cell body and its processes, the axons and the dendrites. All three parts are concerned with the process of conduction of depolarization, जिसे हम कहते हैं conduction of impulses. The cell body, आपको पता है ना, ये स्ट्रक्चर मैं पढ़ करा चुका हूं। सो ये सेल बॉडी है, उसके यहाँ पे छोटे छोटे प्रोसेसेस हैं which are the dendrites, and then there is a long process which is known as axon. Should these processes become separated from the cell body as the result of disease or any simple trauma, they will quickly degenerate. यानी ये तीन चीजें, ये डेंड्राइट्स, ये सेल बॉडी और ये एक्सॉन, अगर यहाँ कोई ब्रेकेज आ जाए, टूट जाए, ये सिवियर हो जाए, डैमेज हो जाए, या डेंड्राइट्स डैमेज हो जाए, तो ऑल दीज कंपोनेंट्स विल डीजेनरेट। यहाँ रीजेनरेशन नहीं होगी, degeneration होगी। ओके, this would explain the necessity of the transport of macromolecule down the axon from the cell body, and also emphasize the dependence of the axon on the cell body. So this axon is obviously connected to the cell body, and if it is disconnected from the cell body, it will die. Okay, now the rate of exoplasmic transport is insufficient to satisfy the release of transmitter substances at the nerve terminal. This problem is resolved in two ways. First, enzymes are present within the nerve terminals in order to synthesize the transmitter from the amino acid derived from the extracellular fluid. And second, at some terminals the transmitter is reabsorbed back. अब ये क्या बात हो रही है? देखें, यहाँ ये बात हो रही है कि we have a neuron जिसका स्ट्रक्चर इस तरह से है कि there is a cell body and then there is axon. अब सेल बॉडी जो कि यूजुअली साइटोप्लाज्म है, यहाँ पर सारी मशीनरी है फॉर प्रोटीन सिंथेसिस। तो न्यूरोट्रांसमीटर अगर यहाँ सिंथेसाइज हो और वो ट्रांसपोर्ट किए जाएं एक्सॉन से, सो दैट विल बी अ टाइम कंज्यूमिंग प्रोसेस, क्योंकि यहाँ बनना और फिर नीचे ट्रांसपोर्ट होना। नर्व इम्पल्स ट्रांसमिशन के पास इतना टाइम नहीं है। नर्व इम्पल्स ट्रांसमिशन हैज टू बी वेरी वेरी क्विक, फौरन ट्रांसमिशन होनी है, क्योंकि जो नर्व टर्मिनल है इसके दूसरे एंड पर या तो कोई न्यूरॉन होगा इसके साथ कनेक्टेड, या फिर कोई एंड ऑर्गन होगा, सो दैट एंड ऑर्गन हैज टू बी सप्लाइड। तो अब ये जो इतना टाइम लग जाएगा, अगर न्यूरो ट्रांसमीटर यहां बनता फॉर एग्जांपल सेल बॉडी में और नीचे ट्रांसपोर्ट होता, सो इट्स गोना टेक सम टाइम। तो नेचर ने इसका इलाज ये किया है कि एक्सॉन के एंड पर यहाँ एंजाइम्स मौजूद हैं और एक्स्ट्रा सेलुलर फ्लूइड से जो भी चीजें उस न्यूरोट्रांसमीटर के बनने के लिए चाहिए वो एब्जॉर्ब होती हैं, और इसी एरिया पर न्यूरोट्रांसमीटर की सिंथेसिस हो जाती है। ओके, सो दैट्स द मैकेनिज्म। तो ये जो ट्रांसपोर्ट में टाइम लगना था वो प्रोसेस अब खत्म। और दूसरा एक इफेक्टिव मैकेनिज्म ये है कि जो न्यूरोट्रांसमीटर यहाँ रिलीज होता है वो दोबारा रीअपटेक हो जाता है इसी पर्टिकुलर रीजन में, ताकि उसको फिर यूज किया जा सके। तो ले दे के सारा मुद्दा ये है, सारी बात ये है कि जो न्यूरो ट्रांसमीटर से रिलेटेड ट्रैफिकिंग है वो यूजुअली इस नर्व टर्मिनल पर हो रही होती है, सेल बॉडी पे नहीं हो रही होती। ओके, सो दैट्स व्हाट इट इज एक्चुअली टेकिंग। Discussion is taking this direction in this particular paragraph. So दोबारा एक दो पढ़ लेते हैं कि this will explain the necessity of the transport of macromolecules down the axon from the cell body, and also emphasize the dependence of the axon on the cell body, and then the rate of the exoplasmic transport, यानी जितना ट्रांसपोर्ट यहाँ से हो रहा है, it is insufficient to satisfy the release of the transmitter substances. तो उसका दो हल निकाले हैं। पहला कि जो enzymes हैं they are present within the nerve terminals, and the second is द फैक्ट नंबर 2 कि इसी टर्मिनल पर रीअपटेक होता है न्यूरोट्रांसमीटर का, विच इज देन रीयूज्ड अगेन. Okay, neuroglial cells, in contrast to the neurons, are non-excitable. These are the supporting cells. They cannot be excited. इनका काम नहीं है न्यूरोट्रांसमीटर को रिलीज करना या depolarization की वेव को कंडक्ट करना. They are smaller than neurons and yet outnumber them 5 to 10 times. They are more numerous than the actual neurons. Okay, they comprise about half of the total volume of the central nervous system. So this is the basic general consideration about neurons and neuroglia. Now, neuronal injury अगर होती है तो उसका reaction क्या होता है? The first reaction of a nerve cell to injury is loss of function. Whether the cell recovers or dies will depend on the severity and the duration of the damage. अगर बहुत severe damage है तो it will obviously die and there will be no regeneration. And if death occurs quickly, such as in few minutes from lack of oxygen, no morphological changes will immediately be apparent. Morphologic evidence of cell injury requires minimum 6 to 12 hours of survival. यानी अगर immediate death हो रही है तो उसमें कोई abnormal features microscopically नजर नहीं आएंगे. लेकिन अगर बहुत ही सीवियर किस्म का स्टिमुलस नहीं था या बहुत ही सीवियर किस्म का डैमेज नहीं था तो दे विल टेक फ्यू आवर्स टू सर्वाइव और कुछ घंटों के बाद उनकी डेथ होगी. तो इस दौरान फिजिकल चेंजेस माइक्रोस्कोपिकली अपीयर हो सकते हैं. The nerve cells become swollen and rounded off. The nucleus swells and displays towards the periphery, and the nasal granules become disappear. So these are all signs and symptoms, microscopic, of cell injury. At this stage, the neuron could recover. If the kind of neural injury were not so severe to cause death, the reparative changes would start to appear. ये जो changes यहाँ सारी बताई हैं, and the cell can be at a point जहाँ से वो दोबारा healthy हो सकता है. Otherwise, अगर स्टिमुलस डैमेजिंग स्टिमुलस परसिस्ट करेगा, हाइपोक्सिया परसिस्ट करेगा, तो इन चेंजेस के बाद देयर विल बी अल्टीमेटली डेथ. व्हेन सेल डेथ इज इमिनेंट और हैज जस्ट अकर्ड, द सेल साइटोप्लाज्म स्टार्ट स्टेनिंग डार्क इन कलर विद बेसिक डाइज एंड न्यूक्लियर स्ट्रक्चर्स बिकम अनक्लियर. द फाइनल स्टेज occurs after cell death, जिसमें cytoplasm becomes vacuolated and nucleus and cytoplasmic organelles disintegrate. The neuron now is dissolved and removed by the activity of the phagocytes which are present in the nervous system, or particularly microglial cells अगर हम CNS की बात कर रहे हैं. Okay, right. So this is not different from necrosis or cell death of any other cell. This is what happens if there is an initiating insult. If the insult is so severe, there will be death. If the insult is not so severe, the cell may regain its normal functions. Okay? In chronic forms of injury, the size of the cell body is reduced. The nucleus, cytoplasm show hyperchromatism, and nuclear membranes and those of the cytoplasm, the irregularity. Not important. Just remember the general principles of necrosis, and they also apply to the neuron. Okay? So that is the initial reaction to injury. Now, axonal reaction and axonal degeneration क्या चीजें हैं? Axonal reaction and axonal degeneration are the changes that take place in a nerve cell when the axon is cut or injured. यानी there is no generalized problem. अगर तो बिल्कुल ऑक्सीजन ही खत्म हो गई है, there is hypoxia, तो ओवरऑल न्यूरॉन डैमेज होगा. लेकिन अगर कोई इंजरी ऐसी हुई है, रोड ट्रैफिक एक्सीडेंट हो सकता है कि ये एक्सॉन कट हो गया है. अब जो चेंजेस आएंगे, those changes will be known as axonal reaction or axonal changes. Okay, they start to appear within 24 to 48 hours after the injury. The degree of change will depend on the severity of the injury. ये common sense की बात है, जितना severe injury होगा उतना उस हिसाब से changes होंगे. The nerve cell become rounded off and swollen. Nucleus swells up. कोई भी injury है ना, तो there are signs of injury then. Nasal granule disperse towards the cytoplasm periphery. The changes reach their maximum in about 12 days. In the peripheral nervous system, section of an axon is followed by attempts at regeneration, and reparative changes take place in the cell body. However, the regeneration is never complete because neurons are in G0 phase. Up central nervous system में the degeneration is not followed by regeneration. तो अटेम्प्ट भी नहीं करते ये सेंट्रल नर्वस सिस्टम के जो न्यूरॉन्स हैं. इफ द कॉर्टिकोस्पाइनल ट्रैक्ट फॉर एग्जांपल आर डिस्ट्रॉयड बाय डिजीज, द नर्व सेल्स दैट गिव राइज टू द एक्सॉन्स डीजनरेट एंड डिसअपीयर कंप्लीटली. सो नो होप. सेंट्रल नर्वस सिस्टम इज सो रिजिड. इंजरी हुई एंड सेल डेथ. An important exception to the axonal reaction of nerve cell described above occurs in the nerve cells of the posterior root ganglia of the spinal nerves. If the peripheral axons are sectioned, यानी कट जाते हैं वो, the nerve cells show degenerative changes. If, however, the central axons are sectioned or destroyed by disease, उनकी examples भी हैं, such as stapes dorsalis, the nerve cells show no degenerative changes. So, general principle ये है कि जो peripheral nervous system है, उसमें अगर axonal injury होगी या axons कट होंगे, so there will be attempts of regeneration. लेकिन अगर central nervous system में damage होता है, so usually the cell bodies and the whole axon system dies. But here they have discussed a special scenario for a particular type of group, which is the axons present in the posterior root ganglia of the spinal nerve. So this is an exception to the rule. Okay. Now, axonal transport and disease spread. Rabies, which is an acute viral disease, dog bite से होती है, is transmitted by bite of an infected animal, dog usually. The virus is present in the saliva of the infected animal. Following a bite, it travels to the central nervous system by way of the axonal transport in both sensory as well as motor nerve. So, if this is the person and a dog bites him here, so virus enters the body and reaches the central nervous system by way of the axons. Axons के जरिए travel करता है. The longer the nerve, the longer the duration of the incubation period. Herpes simplex and herpes zoster are also viral diseases that involve axonal transport to spread different parts of the body. Axonal transport is believed to play a role in the spread of polio as well, and that happens from the GI tract to the motor cells of the anterior gray columns of the spinal cord. So the point is कि एक्सोनल ट्रांसपोर्ट हमेशा जब हम बात करते हैं, सो वी थिंक अबाउट कि सेल बॉडी है और इसमें कुछ सब्सटेंसेस बनेंगे और वो नीचे की तरफ ट्रांसपोर्ट होंगे, बट समटाइम्स द एक्सोनल ट्रांसपोर्ट इज आल्सो इन द रेट्रोग्रेड डायरेक्शन. सो फॉर एग्जांपल, ऑल दीज डिजीजेस दे रीच टू द सेंट्रल नर्वस सिस्टम बाय वे ऑफ एक्सॉन्स. देन देयर आर, I mean, this is not very common, but they have been reported. Neurons के tumors. Recall that the nervous system is made up of mainly of different types of cells, including neurons, neuroglia, blood vessels, Schwann cells, and so many other cells. And each cell, whenever we have cell in any place of the body, there are chances of getting tumor because cells can go bankrupt and they start proliferating quite a lot, leading to formation of tumors. For example, neuroblastoma occurs in association with suprarenal gland. It is highly malignant and occurs in infants and children. So these are the neurons in the suprarenal gland. Ganglioneuroma occurs in the suprarenal medulla. Pheochromocytoma in the suprarenal medulla as well. And that's a very common cause of hypertension because it releases a lot of norepinephrine and epinephrine. Synaptic blocking agent. Transmission of nerve impulses across the synapse is accomplished by release of neurotransmitters. ये बात आपको पता है. Transmission occurs in one direction only because if this is the cell body, neurotransmitters are only released in one direction, down the axon. The released transmitter then exerts its effect on the postsynaptic membrane. Now, this postsynaptic membrane can be another neuron or it can be a muscle or it can be any effector organ. So, ये सब बातें ऐसी हैं जो हम already discuss कर चुके हैं. And then you know what is a synapse. It's a region where transmission is actually happening. So दो neurons for example जहाँ मिल रहे हैं, एक न्यूरॉन ये सेल बॉडी है, ये उसका एक्सॉन है, दूसरी न्यूरॉन की सेल बॉडी है, ये उसका एक्सॉन. So this area where they meet is known as a synapse. As a general rule, long chain neurons with multiple synapses are more easily blocked than shorter ones. General anesthetic agents obviously इस एरिया को ब्लॉक कर सकते हैं, are effective because they have the ability to block the synaptic transmission. In autonomic ganglia पर preganglionic fibers enter the ganglia and synapse with the postganglionic sympathetic or parasympathetic neurons. The nerve impulse on reaching the termination of the preganglionic fiber brings about the release of acetylcholine, which excites the nervous impulse in the postganglionic neuron. So that's अगर हम autonomic nervous system की बात कर रहे हैं, so we know there is a preganglionic fiber, then there is a postganglionic fiber, and again their meeting point is known as a synapse. So no exciting story there, right? Now ganglionic blocking agents may be divided into three groups. So अगर कोई ganglion पे blocking कर रहा है, pre-ganglionic और post-ganglionic transmission को block कर रहा है, so they can be of three different categories. The first group includes hexamethonium and tetraethyl ammonium salts. resembles acetylcholine in the postsynaptic membrane. Thus, these agents inhibit the transmission across a synapse. तो ये पहला ग्रुप है जो acetylcholine को मिमिक करता है और ये एक न्यूरॉन होगा, ये दूसरा न्यूरॉन होगा. यहाँ ये कंपाउंड्स हेक्सामेथोनियम और टेट्राइथाइल अमोनियम सॉल्ट्स ये जाके बाइंड करेंगे. पोस्ट गैंग्लियोनिक न्यूरॉन को लगेगा मुझ पर एसिटाइलकोलिन ने बाइंड किया है, और एक्चुअली एसिटाइलकोलिन ने बाइंड करना था लेकिन अब वो नहीं कर रहा, और उसके बाइंड न करने की वजह से जो भी एक्शन एसिटाइलकोलिन का था वो अब नहीं होगा. सो दैट्स द फर्स्ट कैटेगरी, दे आर एसिटाइलकोलिन मिमिकिंग एजेंट्स. द सेकंड कैटेगरी, which includes nicotine, has the same action as acetylcholine on the postganglionic membrane, but these agents are not destroyed by the cholinesterases. So that's the difference, okay? This results in prolonged depolarization of the postsynaptic membrane. So it persists, nicotine persists for a long time, and therefore it is insensitive to further stimulation by acetylcholine. So actually acetylcholine ने काम करना था, उसकी जगह nicotine लग गया. Unfortunately, this depolarization block is associated with initial stimulation, so these drugs are not suitable for clinical use. So they're not routinely clinically used these days, okay? And the third group of agent which block the ganglia include procaine, which inhibits the release of acetylcholine. So you see, जो पहला group था, first group, that was behaving like acetylcholine. The second group was also behaving like acetylcholine, nicotine. And the third group is blocking the release of acetylcholine. So हर ही चीज में what we are doing is we are blocking acetylcholine ताकि इस गैंग्लियन पर एक्शन ना हो. So this is why they are called ganglionic blocking agents. Okay? In the central nervous system, demonstrating the release of particular transmitter substance at a specific synapse is difficult due to inaccessibility. For example, perfusing a specific localized brain areas through their vascular system is impossible. So here they are actually talking about the प्रैक्टिकल डिफिकल्टी कि किस न्यूरॉन के साइनेप्स पर क्या रिलीज होता है. अब तक हमारी जो अंडरस्टैंडिंग है वो बहुत सारे एक्सपेरिमेंट्स की वजह से, ऑब्वियसली, जैसे हमें पता है आज के व्हाट इज एसिटाइलकोलिन, वी नो हाउ डोपामिन वर्क्स, वी नो अ लॉट ऑफ न्यूरोट्रांसमिटर्स, बट इमेजिन ये कितना डिफिकल्ट काम है कि एक न्यूरॉन दूसरा न्यूरॉन साइनेप्स पे क्या रिलीज हो रहा है, इसको प्रैक्टिकली स्टडी करना बहुत मुश्किल है. ओके So the non-uniform concentration of the norepinephrine in the CNS have led many investigators to believe that it might function as a central neurotransmitter as well. So आज तक हमारी जो understanding norepinephrine, epinephrine और बाकी सारे neurotransmitters की है वो fully developed नहीं है. There are still theories कि maybe this is also working in the different areas of the brain. Dopamine is found in very high concentrations in CNS and secreted by the neurons in substantia nigra. So it is supposed to play a very important role. What? We exactly don't know. Many of the cholinergic blocking agents used in the peripheral nervous system have little or no effect in the central nervous system because they are unable to cross the blood-brain barrier. So whatever is acting in the periphery does not necessarily mean it will also act in the central nervous system. Atropine, scopolamine, and diisopropyl phosphorofluoridate, DPF in short, can effectively cross the blood-brain barrier. So remember these names, okay, because they can get into the central nervous system. Similarly, many psychotropic drugs are believed to affect the CNS activities by influencing the release of catecholamines at the synaptic site. So they cannot enter the CNS, but they can manipulate the activities of different things. The phenothiazines, for example, are thought to block dopamine receptors on postsynaptic neurons. So here, what they're actually talking about are different blocking agents which different sites पर जो जाकर के synapse को block करते हैं. So this is what we have been discussing. Ganglion पे blockage हमने बात की. We are now talking about कि how different blocking agents are only working at the peripheral nervous system and how some of them are entering into the central nervous system. So all important information for your exams, okay? Now, treatment of neurologic diseases by neurotransmitter manipulation. The increasing number of neurotransmitters being discovered in the central nervous system. So look at this statement, for example, the increasing number of neurotransmitters being discovered. इसका मतलब ये है कि central nervous system में बहुत सारे neurotransmitter अभी भी discover हो रहे हैं, okay? And the location of their site of action are raising the possibility that certain diseases can be modified by administration of specific drugs. Huntington chorea, for example, involves loss of neurons that use GABA. So how about using GABA to restore the disease, So this particular disease affects GABA and acetylcholine neurotransmitters. GABA is unable to cross the blood-brain barrier, but physostigmine, a cholinesterase inhibitor, can cross. It can cross the blood-brain barrier, and its use has brought about some improvement in these patients. The use of L-DOPA in the treatment of Parkinsonism has been almost similarly successful. In this disease, it replaces the deficiency of dopamine. So, अगर हमारी अंडरस्टैंडिंग न्यूरोट्रांसमीटर्स की बढ़ती जाएगी कि कौन से न्यूरोट्रांसमीटर्स कहाँ रिलीज होते हैं, किस डिजीज में कौन सा न्यूरोट्रांसमीटर कम होता है, इफेक्ट होता है, अगर ये अंडरस्टैंडिंग डेवलप होगी तो ऑब्वियसली वी कैन यूटिलाइज many drugs for the treatment of such disorders. Drugs are now rapidly being developed to modify the process of synaptic transmission in a number of ways. For example, by interfering with the process of neurotransmitter synthesis, by inhibiting the uptake, by binding to the neurotransmitter, and by terminating the neurotransmitter action. So, multiple तरह से neurotransmitter को block किया जा रहा है, उनके functions को augment किया जा रहा है. अभी भी मैं आपसे बता रहा हूँ अपने experience से कि हमारी understanding about the nervous system is very very poor, very initial stage पर है. इसीलिए हमें जितना ज़्यादा समझ आएगा उतना ज़्यादा हम काम कर सकेंगे. And therefore, this is a very active area for research. Now, neurological reaction to injury. The reaction of the neuroglial, not neurological, I misread it. It's neuroglial. अब तक जो हम बात कर रहे थे, we had been talking about neuronal. आपको पता है ना नर्वस सिस्टम में ये पूरा चैप्टर क्या था? This is chapter number 2. Chapter number 2 is about neuron and neuroglia. So first we talked about what happens in the neuronal injury, and now we are talking about what happens in the neuroglial injury. Okay, so this particular heading now is talking about neuroglial injury. So the reaction of neuroglial cells to injury, whether caused by physical trauma or vascular occlusion, is characterized usually by hyperplasia and hypertrophy of the astrocytes. यानी जब इंजरी होती है तो ये अपना नंबर बढ़ा लेते हैं. द प्रोलिफरेशन ऑफ द एस्ट्रोसाइट इज रेफर्ड टू एज एस्ट्रोसाइटोसिस और ग्लाइयोसिस. सो अगर ग्लाइयोसिस हो रही है या एस्ट्रोसाइटोसिस हो रही है, यानी नंबर ऑफ एस्ट्रोसाइट्स बढ़ रहे हैं, दैट बेसिकली इंडिकेट कि कोई ना कोई न्यूरोग्लिया के लेवल पर इंजरी है. The loss of neuronal tissue is not compensated for in volume by the glial hypertrophy. The cytoplasm of the enlarged astrocyte contain large number of fibrils and glycogen. The dense feltwork or astrocytic processes that occur in the areas of neuronal degeneration are called glycotic scars. So basically, जैसे किसी और टिश्यू में, लिवर में या किसी और टिश्यू में जहां इंजरी होती है, इन्फ्लेमेशन होती है, उसके बाद स्कार बनता है, इसी तरह अगर न्यूरोग्लिया की इंजरी है तो ग्लायोरिक स्कार्स बनते हैं. ओके, दिस इज व्हाई इन पेशेंट्स विद फोकल एपिलेप्सी ड्यू टू लार्ज ग्लायोरिक स्कार्स द स्कार इज एक्साइज्ड सर्जिकली. वो मतलब इतना बड़ा भी फाइब्रोटिक टाइप का स्कार बन जाता है कि इट हैज टू बी रिमूव्ड सर्जिकली. Oligodendrocytes respond to injury by expanding and showing vacuolation of their cytoplasm. The nuclei also tend to become pycnoric. So again, these are markers of injury. Microglial cells in inflammatory and degenerative lesions of the CNS retract their processes and migrate to the site of lesion. So these are kind of the neutrophil जो move करके पहुंच रहे हैं, macrophages. Here they proliferate at the site of insult and are actively phagocytic in nature. They try to take away whatever damage has been done. उसको खा पी के phagocytose करके खत्म करते हैं. Microglial cells are active in number of diseases such as multiple sclerosis, AIDS, Parkinson's disease, Alzheimer's. So these are all conditions which in some way or the other have some either inflammatory component or some damage component जिसकी वजह से microglial cell activate होते हैं. जो neuroglial cells हैं, since they are also cells, they may have some tumor conditions, neuroglial tumors. Tumors of the neuroglia account for about 40 to 50% of the intracranial tumors. So that's not uncommon. That's actually very common. Such tumors are referred to as gliomas. Okay. Tumors of astrocytes are those most commonly encountered, and they are examples हैं astrocytoma and glioblastoma. Apart from the ependymomas, tumors of the neuroglia are highly invasive. So इनका metastatic potential बहुत ज़्यादा है. Therefore, they are very lethal. This explains the difficulty in achieving complete surgical removal, because आप इस जगह से अगर well circumcised कोई चीज होगी तो उसको remove करना आसान है, लेकिन वो अगर invaded है तो उसको remove करना बहुत मुश्किल है. Another feature is that as these tumors infiltrate, they often do so without interfering with the function of the neighboring neurons. As a result, the tumor can be very much larger than the symptoms and the physical signs. So because ये astrocytes के tumor हैं, बराबर में जो neurons हैं, they may be fine. So these cells are proliferating, but the neurons are okay. So function of the brain is okay. तो काफी बड़ा tumor होता है, लेकिन बंदा बिल्कुल ठीक होता है. That's not uncommon. Multiple sclerosis is one of the most common CNS disorders affecting about 2.5 lakh Americans. ये पुराने stats हैं, अब number बढ़ गया है. It is characterized by the appearance of patches of demyelination in the CNS. So myelinated areas disturb होते हैं, demyelination patches, okay? The myelin sheet degenerate and the myelin is removed by microglial cells. Astrocytes also proliferate, leading to formation of a gliotic scar. As demyelination occurs, the conduction of the nerve impulse is obviously affected. Because raising the temperature shortens the duration of the action potential, one of the early signs of the multiple sclerosis is the symptoms and signs that can be improved by cooling. So if the person says, I have some issues, but then, by cooling, I'm okay. By hot bath, I'm not okay. In the summer area, I'm not okay. There may be early signs of multiple sclerosis. Most cases occur between these ages. So see, it's pretty young, 20 to 40. The cause of the disease is unknown, although an interplay between a viral infection and a host immune system may be responsible, but the pathogenesis is not truly understood. Then we have to discuss what is cerebral edema. So edema is obviously accumulation of fluid. अगर वो CNS में है तो it is cerebral edema. It's a very common clinical condition that can follow head injury. सर पे अगर चोट लगे, cerebral infections या फिर brain tumors. The resultant swelling of the brain may lead to flattening of the cerebral gyri, herniation of the brain that can cause even death. अगर foramen magnum से brain stem निकल गया तो death भी हो सकती है. So herniation, because mass बढ़ रहा है ना, if this is your brain, if it is accumulating liquid, it will herniate because उसका mass बढ़ रहा है. So it's a serious condition. It is an abnormal increase in the water content of the central nervous system and takes 3 forms. Either it can be vasogenic edema, is the most common type and is due to accumulation of tissue fluid in the extracellular space following damage to the vascular capillary. So their permeability increases and there is edema. It can be cytotoxic edema due to accumulation of fluid within the cells of the nervous tissue, resulting in cellular swelling. So यहाँ extracellular space में fluid जमा नहीं हो रहा जैसे vasogenic edema में हो रहा था. यहाँ nervous cells जो हैं, neurons and glial cells, वो swell हो रहे हैं, उसकी वजह से edema हो रहा है. The cause may be any toxin or any metabolic problem or any sodium-potassium disturbances. Then there is interstitial edema occurs in the obstructive hydrocaph when there is rise in the cerebrospinal fluid pressure, which forces the fluid out of the ventricular system into the ECF. So either it is because of the increased permeability of the blood vessels, or it is because of the swelling of the neuronal cells, or it is because of the blockage of the CSF flow, जिसकी वजह से CSF push out होता है into the spaces, extracellular spaces of the CNS. In तीनों ही conditions में there will be cerebral edema and this is life-threatening. 2 anatomical factors must always be remembered in edema. Number 1, the brain volume is restricted by surrounding skull. So brain की बढ़ने की एक capacity है ना उसके volume की, क्योंकि इर्द गिर्द बहुत सख्त किस्म का skull है. And then the tissue fluid is drained primarily into the venous sinuses by the cerebral veins because there is no lymphatic drainage. So no lymphatics in the CNS is an important point. And both these points are something जो अक्सर स्टूडेंट्स भूल जाते हैं. Now इनका impact क्या है? इनका impact ये है basically कि because there is a very rigid skull outside, और अगर brain का size बढ़ रहा है, so the brain has no option but to herniate, and if this herniation happens, it can lead to death. And this is why brain edema or cerebral edema is very, very serious stuff. Okay, so that's all about the clinical stuff associated with chapter number 2 of Snell neuroanatomy. चलो आपसे मिलूंगा अगली वीडियो में. अपना ख्याल रखना.